The format of today's overview will essentially consist of three different items. There's going to be a brief DNASTAR introduction. We'll actually do a live demo of the new automated cloning interface in our SeqBuilder application, and then a question and answer period at the end. As Sharon mentioned, feel free to submit any questions to Sharon directly via that live chat, and we can answer them accordingly. Why DNASTAR? DNASTAR has a solid 30-year track record of developing, supporting, and responding to the needs of today's researchers. For example, we have always been among the first companies to support new data types, trends, and workflows. This continual development has created today what we call our DNASTAR Lasergene suite. DNASTAR Lasergene is an integrated solution of bioinformatic tools for molecular, genomic, and structural biology. For example, within the suite, one can perform diverse workflows such as primer design, annotation, and cloning in our SeqBuilder application, genome assembly, variant calling, our seqment engine tool, and structure, function, and ligand binding prediction with our NovaFold. We take great pride in creating easy-to-use solutions for today's life science researchers. We feel that we've created a comprehensive set of tools that can handle any workflow in virtually any platform. This focus is quite evident in how our software is organized, both scientifically and logically. DNASTAR's core values center around essentially four focus areas. Accuracy. We take pride in creating time-tested, accurate solutions. For example, for our next-gen sequencing data, we critically test our algorithms with Illumina Platinum and Genome in a Bottle consortium standards. We take great pride in not just getting an answer, but getting the correct answer. This accuracy is a result of our highly vetted development and quality assurance practices. Our attention to detail. And over 30 years of experience in this field has resulted in a tool that is widely accepted all over the world. We are proud to be identified as the most widely cited bioinformatics software in peer-reviewed publications. At last count, our solutions have been cited in over 52,000 instances. Trusted global outreach. As a company, our reach extends globally. We have customers in over 90 countries and are present in virtually every major academic, government, or private research institute. Following are some of the exciting projects where our software was utilized, whether tracking the lineage of King Tut back numerous generations with the Egyptian Museum, or being on the front lines in today's Ebola or tuberculosis research. Our solutions are easy to learn, logical, and developed by scientists for scientists. We focus on integrating our solutions whether by data type, workflow, or across platforms. This simplicity is evident with our modest hardware requirements and support resources and tools we have created to assist users with the software. For example, our DNASTAR Navigator interface is a helpful tool where researchers quickly find help resources at the click of a mouse. This support is quite evident directly on our website. Since most of you probably heard about our webinar, whether from a mailing or directly on our website, it's quite evident that we have numerous webinars that we've created over the years, numerous videos that we've created over the years. And then also, if you simply have a question, you need a real-time answer, you can chat with us live directly on our website to answer a question or a specific recommendation for a project. Lasergene 14 would be the most current release from DNASTAR. This release expands on our molecular, genomic, and structural biology tools to highlight some of the key enhancements. In SeqBuilder, we have developed new tools for automated cloning, whether using Gibson, InFusion, GeneArt, traditional restriction enzyme digest. We've incorporated the new NovaDoc tool, which incorporates protein docking into our software. The tool is powered by the SwarmDoc algorithm from Cancer Research UK. We've created the new NovaFold antibody workflow for modeling and antibody development. And then we've created a new interface called GeneVision Pro for integrated genomic analysis. Today our focus will be on the SeqBuilder application, which is in our molecular biology suite. The molecular biology suite would be the configuration that would provide you with the common foundational tools for annotation, primer design, cloning, restriction enzyme analysis, Sanger assemblies, pairwise and multiple sequence alignments, blasting, and just to name a few functionalities. SeqBuilder would be our flagship application for sequence editing, annotation, primer design, plasmid map creation. The addition of the automated virtual cloning tools is a strong addition to the application. We have essentially created a tool that makes cloning as easy as 1, 2, 3, where 1 is preparing your insert, 2 is selecting your vector of interest, and 3, obtaining results. This can all be done in a matter of seconds. Let's jump into the software to show the enhancements. Once SeqBuilder is open, you will need to add your target sequence into the application. This can be done easily by a number of different ways. By selecting File, Open, Sequence. What you would do here is you would simply navigate to your sequence of insert that you want to add into SeqBuilder. As you can see, there's a number of different file types that Lasergene does support, whether standard GBK files from Ncbi, EMBL, FASTA files, or DNASTAR Lasergene files. Another option to bring in your data would be from File, Open, Entree Sequence. This selection will allow you to pull a selection directly from Ncbi, where you can specify a database. So for example, if I simply select 1, 2, 3 here, what it's going to do is automatically connect me to Ncbi, prompt me to save this file to my desktop or on another folder of interest, and it will automatically open that sequence directly in the application. One key advantage of working and pulling data directly from Ncbi is a couple things. One, you'll pull over annotations as well as citations that are linked to this project. And then I can actually turn these annotations on directly in the software for further downstream analysis. Another option to open the data is simply dragging, dropping your file directly into the software. So I have another monitor here, so I'll drop a sequence into SeqBuilder. And as you can see, the file opened up readily. Once the insert has been added, we can proceed with using the target in our automated cloning workflows. To start, we first need to highlight the nucleotide string that you would like to clone with the cloning interface. The software will automatically amplify the insert with the appropriate overhang suitable for the cloning technology that you specify. To select a nucleotide, one can simply highlight a region of interest. This would be suitable if you're working with a FASTA file and all you have is basically sequence coordinates. Another option is you can apply different open reading frame filters. So on the left-hand side under the views, there is actually an open reading frame view where you can toggle between top strand and bottom strand. What this would allow you to do is you can take into consideration the frame, the presence of initiating methionines, transcription stops, open reading frame size, and so forth. Or the easiest option, since I'm working with an annotated file here, is I can simply turn the features on, and all the features that are associated with this file would automatically be shown. Once the target has been highlighted, in this specific case, I'll just highlight the potassium channel protein here. Once the sequence target has been highlighted, we will click on the cloning pull-down to select the cloning technology of interest. So from the cloning pull-down, as you can see, there are a number of automated cloning workflows that you can select. Upon selection of the cloning workflows, the default parameters appropriate to that technology will be selected. These parameters can be changed, if needed, further downstream. In this specific case, let's simply just select Gibson LAC cloning. Upon selection of the cloning technology, a cloning project window will automatically populate. This window will act as an interface for the cloning project and track target, vector, and result steps for the project. The window is divided into essentially 3 parts. There's the project tab. So this will keep a running track of what inserts you're adding, what vectors you're utilizing, and the resulting clones that will be propagated and the targets that will be amplified. The clone tab here is where you can name your project, specify what cloning method you want to utilize, change what preferences, default preferences or parameters for that cloning project, as well as select a vector of interest. Built into the software, there's actually a number of different cloning vectors that we've already supplied with the software. So, for example, the T vectors, PGEM, PBR322. But the software does also give you the capability to add your own vectors of interest, whether specific to a vendor that you like to utilize or a workflow. And then there's a third tab called the Summary tab. This is basically going to keep track of the fragments that are generated, the approximate size of the clone, the appropriate steps for that protocol, as well as taking a look at the fragment junctions where you can look at the top strand, bottom strand, as well as all 6 frames to make sure that your insert is in the correct frame from the promoter elements. Within the Clone tab, there are a number of methods that you can select. From this window, you can select a specific cloning technology, and the underlying parameters would be optimized accordingly. For example, when selecting the preferences for Gibson Assembly, the default parameters can be changed specific to your project. For example, some Gibson Assembly workflows would require a 40 base pair overlap instead of a 20 base pair overlap here. So you could change that here specific to whatever workflow or primer concentration or minimum primer length that you need to specify for your project. For this project, what we're going to do is we're actually going to add a Gfp protein, a green fluorescent protein, upstream of our target. So what we're going to do is I'm simply going to drag and drop a sequence file, add that to my inserts folder, and then what we're going to do is I'm going to drag that Gfp protein into the fragments. And since I want my protein to be downstream of the GFP, I'm going to change the order of that fragment. As the interface tells you right now, now we need to choose a vector. So what we will do is under the vector pull down, we will select a pgem vector. So I'll scroll up here, select pgem, and I'll simply take the first pgem vector. The next step is it's going to give me the approximate clone size here, but now we need to linearize the vector. So I'm going to select linearize. And what's going to open up directly is the pgem vector itself, the expression vector. What you need to do now here is you need to select the site in which you want to clone into. As you can see, there's a number of different options here. It will give you the unique cutters for the restriction enzymes. If this was a restriction enzyme project, I could specify, for example, if I wanted to use EcoR1 5', Indy3 3', I can directly make that selections directly in the software or directly in the interface down here. Since we're using Gibson Assembly here and restriction enzymes are not utilized, I'm simply just going to use the same site in which EcoR1 and Indy3 are going to cut. So I do know that EcoR1 cuts here, 5', and it will extend all the way to roughly right here, 3'. Once I've selected my region of insert that I want to insert my target, I'm going to proceed to select the select button here down at the bottom. Upon selecting there, as you can see, the GFP is present 5', and that my DAG.sequence is present just downstream of that DAG sequence. From here, I'm going to select try it, and what's going to happen is the cloning summary window is going to populate with the fragment size, the approximate clone size, the primers suitable for that reaction, and the steps you would take to correctly amplify the vector, as well as the 2 targets that I've selected for my experiment. At the bottom here, there's going to be the fragment junctions. So this is a tool in which you can start to assess the frame, the transcription stops, and just to make sure everything is in the correct frame and the orientation for downstream assembly alignment. If all of this looks suitable to you, the next step is you would simply make it. And what populates in that cloning project window is the amplified Gfp protein, the amplified target DAG protein, and then the actual resulting clone where both of those inserts have been inserted into the expression vector. Let's simply just take a look at one of these amplified inserts. So let's just double click on NC011521 GFP. What will open up is that amplified inserts. You can see that 5', there's the primer that's suitable for amplifying the ends. 3', there's the other primer for suitable creating the overlaps on the 3' end. As well as we're keeping track of the primer list as well. Should you want to further enhance these primers, look at hairpin structure, dimer formation, and so forth, you also do have the capability to work on this primer. So you'd simply click on one of the primers and actually go to our priming work on primer interface. And this will give you false binding site, hairpin information to further manipulate or enhance this primer suitable for your workflow. Let's close out of this and actually show you the resulting clone. So we're going to double click on this clone. What's going to pop up by default is the sequence view. At the bottom here is that cloning summary again. So if you want to visualize the summary, I can drag this window up, scroll down, and there's my primers, my steps, my fragment junctions that I can utilize for this cloning strategy. If I want to visualize this in a linear or a circular map view, you have the ability to toggle between the different views, so whether linear map or say I want to create a circular map, you do have that capability in the software. So as you can see, if I turn all these features on, there's my GFP, there's my insert, and you have the capability to manipulate this image, hide, change styles as you see fit. All right, so let's close out of this project. And I just wanted to show you how to automate this process even a little bit faster. So let's just simply perform a GeneArt project without all the talking. So what we're going to do...
Actually, Brian, can I interrupt you just for a moment?
Sure.
I have a couple of questions that have come in about the Gibson cloning workflow. The first question is, you made a selection when you were linearizing that. And can you also use restriction enzymes? How would you go about making a selection based on restriction enzymes instead of just dragging your mouse across?
Yeah, so let's just go back to that project there. So what I'll do is, let's just amplify this insert. Let's select all. We're going to go back into the cloning, Gibson LIC cloning interface. So what you would do basically, let's go back to PGEM. You can linearize. So what I just basically did is I highlighted a selection that I knew it's going to clone into. I knew that that's what the cut site was. I knew that's going to be in frame of that T7 promoter that's upstream. If you simply want to select two restriction sites, you can do that as well directly in the sequence view, or down here in this newer interface that we've created, I can simply select my left cutter. So Echo R1, my downstream, my 3' cutter, so Hindi 3, and then you would simply select.
Okay. Thanks. There's actually another question that also is about that that display, which is what determines which restriction enzymes are displayed when you're doing that linearization?
Okay, sure. What we're showing here is actually the unique cut sites that are present in the target. SeqBuilder does also allow you to apply different selectors, enzyme selectors, and so forth. So if I, for example, if I expand, let me just toggle this over a little bit. If I expand on this enzyme display folder, there's actually a number of different selectors that are built into the software. So if you want a specific enzyme type, like 5' overhangs, 3', blunt cutters, absent sites, you have the capability to create a selector based on whatever criteria that you specify. The selectors that are built in, the enzymes that are built into the software will actually be from rebase catalogs, so New England Biolabs restriction enzyme catalog, but you have the ability to create whatever selectors you utilize. Okay. All right. Cool. Yeah, thanks for the questions. So let's just go back to that original file. And what we're going to do is actually just do a GeneArt automated cloning project without all the talking. So essentially what you're going to start with is you're going to select my inserts. So let's take that potassium channel protein again. I'm going to go to the cloning pull down. I'm going to go to GeneArt cloning. That cloning project interface is going to pop up. If I click on preferences, my overlaps are going to be suitable for GeneArt. My primer length will be suitable for GeneArt. The next step is to select my vector. So I'm going to select that PGEM vector again. I'm going to linearize, and then I'm going to specify that cut site again. I'm going to highlight that selection, that cut site. I'm going to select the select button. I'm going to select try it. I'm going to assess the results. So I'm going to look at the primers. I'm going to look at the steps. And the key thing is to look at the frame to make sure everything is in frame. In this specific case, I'm just doing one protein, one target. But whether you want to do one target, 2 targets, 6 targets, entirely up to you. I'm going to make it. And then I'm going to assess the quality of my results. So what we've done here is automated the process entirely. So all the basic various technologies go through this interface. So in this interface, you simply select the cloning method, and the software, the underlying parameters, will do everything for you to add the appropriate ends. All right. So let's kind of take a look at what we've done with the traditional enzyme digests. Historically, in our software, you previously would have had to prepare your target, amplify the correct primers, 5' and 3' on your target, open another file of the expression vector, and then basically clone that restriction insert into that expression vector of interest to make sure all the restriction sites map up. What we've done is we've automated that whole process entirely as well. So I'm going to close out of this project here, and then we'll go back to that potassium channel protein. So for traditional enzyme digests, you would do the same thing. Highlight your target of interest, select cloning, restriction enzyme cloning. The cloning project interface is going to open up. As you can see, there's my target that's been added. The next step is I want to choose my vector, my expression vector. Let's go back to that PGEM vector again. The next step, as the software is telling you, is I now need to digest it, just like you would in the real world. You need to digest your vector to linearize it. Here, you're going to specify your restriction sites. So let's just select echo R1 again, and let's select the 3' site, ND3, and then I'm going to hit the select button, and let's try it. It's going to automatically give you the clone size, the steps that you would need to take to prepare the inserts as well as the vector, and then your fragment junctions to look at the frame. If that looks suitable to you, you would select Make It. There's your amplified inserts with the appropriate primers to add the 5' and the 3' ends for your restriction sites, and the resulting traditional restriction enzyme clone with the cloning summary reports. If I click on the circular map view here, drag this down, you can see my potassium channel protein has been inserted into that PGEM expression vector of interest. So what we've done here with this new automated cloning wizard is we've essentially made cloning as easy as 1, 2, 3, where 1 is your inserts, 2 is the vector that you need to select, and 3 the final results. What typically took couple of minutes previously, you can do this in a matter of seconds to get your primers and your final clones. Like the new cloning methods themselves, we automate the entire process. Thank you for taking the time out of your day to learn about some of the new solutions. Does anyone out there have any additional questions?
Yeah, Brian, thanks for that presentation. We have 2 more questions that have come in. One is, I see that primers are designed as part of the process, but is there any way to assess the quality of those primers?
Sure, great question. So let's just go back to those amplified inserts that we've created in that project folder. So we're going to click out on this. So this is that restriction project, traditional restriction digest project that we just did. As you can see in this project, there's an inserts folder, there's a vector folder, and then the resulting results of that restriction digest. So there's the amplified inserts and the actual final clone. If we click on this amplified insert here, if I double click on that, what's going to pop up is your 5' and your 3' primers that you've created as part of this automated process. If you want to assess the quality of those primers, what you would simply do is, the easiest way is to go to your primer list, expand the primers that we've created, click on the top strand, under the priming pull down, you'd work on that primer pair, and then what's going to pop up is actually the entire primer that's being created. So what this is kind of showing you is the dimer formation, the stable dimer, as well as the hairpin information. These primers that we created are based on the nearest neighbor method. We try to err on the side of caution just to make you aware of things that you may need to take into consideration in regards to primer design. When we say that there's bad, what we're saying here is there's some results here that you may need to think about that may affect the downstream quality, this primer for whatever reaction you're working with. If you want to further optimize this primer, you do have the capability to introduce a codon change or a mutation where you can do a silent mutation directly in the software to kind of resolve some of these issues. But just because we say something is not optimal here, it doesn't mean the primer won't work. You would need to empirically determine that in the lab. There's numerous instances where I've come across customers that, what we say. So, for example, a T7 promoter primer historically is considered a bad primer. If you look at the nearest neighbor method, the presence of stable dimer, hairpin, stable pair dimers, and so forth. Just because we say there's a result here may need to take consideration doesn't mean the actual primer won't work. So, but yeah, you would need to basically right-click on that primer and then work on that primer pair, and then you could save that accordingly in the project. Once you've created those primers, you also do have the capability to export them. So in that primer list view, you simply click on that view and then File, save your primer catalog, and that you can export that as a FASTA file or a primer catalog file for further downstream use.
Great, thanks for that explanation about how primers work in this cloning workflow. And before we end, we have one more question, and that is, how can I see the expressed protein or product if I want to assess codon usage or size? Oh,
that's a great question. So the easiest way, to do that is, to open the clone, let's go to the circular map. So in our software, you have the ability to do a number of different transformations. So those are available from the sequence pull down, new sequence. So whether you want to do a translation of that selection, complementation, change the origin, whatever, you would simply just highlight that amplified product within that expression vector, and then you will go translate. And then what the software is going to do is it will actually give you the translation at the bottom, but it actually gives you some results or statistics, at the top here in the comments view. So whether molecular weight, isoelectric points, and then if I scroll down here, it will actually give you a codon usage chart. So you can compare this to, a rare codon optimization chart to see if you potentially could have some codons that you need to, for example, use a competent cell that has tRNA for rare codons and so forth.
Okay, Brian, you thought you were going to escape with just that final question, but we actually have one more that just slipped in. Is there any way to set parameters when you're doing the cloning workflow regarding the primers? Can you change their melting temperature, %GC, things like that?
It's a great question. So yeah. So as part of the primer design workflow interface, you have the capability to change parameters. So that's more of a target-specific enhancement. So, for example, if we initially start with that protein again, in the software you do also have the capability to create your own primer pairs, whether create primers from a selection, so you can randomly select a 20-mer and create a top strand and a bottom strand primer, or if you want to create primer pairs of your target, you would simply go create primer pairs, and there's actually an interface that will allow you to set change conditions, primer characteristics, target TM, not GC content per se, but there is some primer optimization options here that would allow you to manipulate or create additional primer options.